Okay, so thank you for the opportunity. I'm going to talk about observational research on this on COVID-19. Hold on. There we are. OHDSI, Observational Health Data Sciences and Informatics, is this international network. Columbia is the coordinating center. About 3,000 people are researchers on it nowadays, with 150 databases and data on about 600 million unique patients from around the world. Not much COVID data yet. It's mostly retrospective data. This past weekend, actually more than weekend, we had an 88-hour study-a-thon with 350 researchers working on COVID to see what we could extract. Most of it was, this was just the kickoff to a longer-term project. So I'm going to show you what we were able to do in the first couple of days, but the research is ongoing. As you can see, the network is around the world there. First thing I want to show you, and I may catch up some time for you, Andrea. First slide just showing the age distribution. Now I know in Korea,
very grateful.
For the social groups that were where it's spread in Korea early, may have biased towards young, but the main bias here is I think that they're just testing everyone. And what we see is that most people are between age 20 and 44. At Columbia, we see the age distribution where most people are between 60 and 80. So what we're seeing here is I think it's an infection of young people and a disease of old people. And that further means that the numbers that we're getting when we look at our measurement numbers, we're seeing just a tiny sample of the actual population in New York. And we all know that, but to see it graphically here is pretty unfortunately impressive. So when we get our numbers for how many we're seeing infected, those are the ones we're testing. The Korean population gives you a feel of the subsample that we're using. We're seeing about equal split, male and female, you can see on the bottom. I'm not going to go through detail on these comorbidities. These are raw rates, so these are uncorrected. So these, of course, are going to be biased numbers. And I'll show you some examples of studies in a couple slides. But what comes right out is one of the biggest ones is hypertensive disorder, and the other one is heart disease. So we are seeing that bias in our population, that the ones who are coming to us and are warranting a test are, in fact, most of them have had hypertensive disease diagnosis. Let me just say that the medication use on the lower right of that screen, that includes their old use and their use in this hospitalization that's mixed together on this slide. So that's why you're going to see some of those things like antibacterials being so high. So one study, now I'm going to show you two studies. Now these may not be turn out to be the most important studies, but we're building up our ability to do this. And so angiotensin pathway we've heard about. So ACE inhibitors and ARBs, angiotensin receptor blockers, the question is people who have been on them, are they favored or are they hurt by having been on that chronically? So we went ahead with the hypothesis and we compared ACE and ARB use to calcium channel blockers in hypertensive patients, and how do they do? And here's our result here. As you can see, actually it's blocked on my screen because let me just move. I don't have to move it. I kind of remember. This is the table is from Korea. You see a hazard ratio of 1.2 is irrelevant because the confidence interval is from 0.1 to 10 basically. So what that's saying is we don't have enough information yet. So we're coming up with new ways to do these causal models. At Columbia, we're not seeing a major detriment for having been on ACE or ARB. Our raw odds ratio is 3 in favor of having been on them. But if you look in Korea, as soon as they started doing just some adjustments, they brought it down to 1.2. So the real lesson from this slide is when you go online and read these papers that do these simple observational studies of you know, what is important and what are the covariates in this disease, you have to be very careful to interpret it because we'll go from an odds ratio from 1 to 3 by just doing simple corrections, and they are not even necessarily doing those simple corrections. Also, let me point out, to study these drugs, randomized trials are the primary means to do it, but where we have observational evidence, we'll use it. The FDA and also the European FDA, which is called the EMA, has asked us for the safety profiles of drugs. So hydroxychloroquine, whether it turns out to be helpful or not, but we're looking at all the drugs, antivirals, DMARDs, et cetera. So this weekend we ran this study looking at outcomes that were important. Remember, things like QT prolongation is important for hydroxychloroquine and so on. So we assembled a sample this weekend of 130,000 patients on hydroxychloroquine and 57,000 on a comparator drug. And shown on the next slide, we were able to report to the FDA we're not seeing any major side effects. Now, when you look at the confidence intervals, there's something like 0.8 to 1.2, 0.9 to 1.2, 0.9 to 1.1. So these are very narrow confidence intervals for these kinds of studies. So the FDA can look at this. Although hydroxychloroquine is an old drug, it's not well studied, its safety profile. So this is the first study they've ever gotten international of this size to have narrow confidence intervals and be robust to tell them whether it's safe. We'll be looking at, we're marching through other drugs as we go. So that was just an update there, and there'll be more to follow as the community moves forward. Now, a little bit about the Columbia Data Mart. So we're assembling with the faculty and fellow mentioned at the top of the screen, the Columbia COVID Data Mart. It's going to be placed in the tri-institutional data warehouse, which is called Jupyter, although we'll keep it separate, Cornell and Columbia, as two different marts for now. What we can gather now are demographics, visits, labs, including the COVID test and medications, and we're working hard on getting the diagnoses, procedures, vitals, and then notes in. Remember that we just switched to Epic 6 weeks ago, and so, or 2 months ago. So all the processes to get out of Epic, unfortunately, are not really operational yet, so we're doing it as we can. How do you get to these data? Well, access is via the usual means, an IRB approval and then a track request. As you can imagine, we have a backlog of requests right now, but we're going through as quickly as we can. The best way to access it, if you have a clinical data navigator. So most departments or many departments have a person identified to do queries against the warehouse. That person can get to the database, so medicine, surgery, et cetera, have a person. And also the CTSA, Murdoch's group, can also help get access. Because we have limited data, we have several manual data extraction projects going on. And I want to thank the medical students. I want to thank the deans for allowing this to happen, are literally logging into the system and typing it into a REDCap system to get these data marts. One of them, run by Ray Chen, is joint with Cornell, doing the basic parameters of the COVID patients as they come in. And then each of the specialties, for example, in ID, is also collecting data, also on REDCap. And we hope to merge it eventually. And many of the subspecialties in medicine are going in with ID, and I know pulmonary, et cetera. And cardiology is merging with RMART. So a lot of people, like 100 people, are working hard to get these data collected to make them available to the Columbia community. And I'll stop there.
Thank you, George. Any question? I see one raised hand. Raul Rabada.
Yes, George. I was wondering if there is any link to genomic data in what you are collecting, or if there is some part of this information has already been typed. I mean, both with the SNPs or sequenced.
The 600 million have not, but there's other efforts to get it linked. You know, for example, our eMERGE database, well, not many, we have our, whatever, 10,000 patients in eMERGE, but they haven't been, oh, by the way, the numbers were, from Korea, we have over 4,000 COVID patients. From Colombia, there are over 1,000, I guess. But among our 10,000 in eMERGE, almost none of them have COVID. So, you know, the All of Us Research Initiative, we're working with them, and we'll see, we do have samples, but I don't know how long it's going to take them to gear up. So there's a couple of parallel efforts, but it's I think the Columbia Biobank is going to be more effective locally. So you've already heard about that. I think around the world, we're looking at a couple of initiatives, but I don't have, it's not part of the Odyssey initiative, Raul.
And Sam Silverstein.
Yes, George, is there a communication network for all New York City institutions when convalescent plasma becomes available? It's going to be important for all New York City institutions to talk to each other.
Okay, so you mean at the level of not at the not communicating data itself, but communicating, coordinating.
Yes.
I think others can speak about that better. I think I would have to go to the dean's office and see how they're communicating this kind of thing. So I won't answer that, but we can find out. There is communication about the data, and that's the PCOR network that we're part of help coordinating. So we're actually trying to get a New York City sample, but that's not as far as what I've told.
No, I just think that this will be very important as you get to something that approaches therapy. The demand will be enormous.
Yes.
Okay. Judith Corner, you're able to ask your question?
Yes, thank you. This may be premature to analyze the typical comorbidities, but I noticed on the slide that you have is that obesity was only prevalent in about 14% of the population, which seems a little odd given the typical rate of obesity at Columbia is certainly over 40%. I was wondering about your thoughts on that.
That's a good question. So my caution is these raw numbers. As I said, for ACE and ARB, the odds ratio went from 3 to 1 with minimal changes. So I'd have to look, but it's a good question. It's a good thought. So with the data, you can actually get to the MART and actually do that analysis. We could try to look also. We're doing many things at once, though. But it is a good question.
Yeah, we'll be putting in a request for that. Thank you.
Good.
Okay, great.